Hello everyone and welcome to Stevanato Group's webinar on innovative solutions in pre-filled syringes for biologics and ophthalmic applications. Hosted by Pharma Demand, my name is Scott and I will be your moderator today. So during this webinar, Enrico Barichello, Product Manager for Syringe Platforms, and Alan Shu, Product Manager for Analytical Services, both from Stevanato Group, will be discussing the best strategy between customers and suppliers to select the right container based on the key features of the combination product. So at this stage, I'll hand over to Alan, who will take the webinar forward.
Thanks very much, Scott. So as he introduced, my name is Alan Shu, and I'll just be briefly talking about Stevanato Group at a glance. So we're most famous for our drug containment solutions. So these are things like cartridges, vials, and syringes, but we also make in vitro diagnostics, drug delivery systems, other engineering equipment, which can include sub-assembly and final assembly inspection equipment. And then where I focus on, which is our analytical services branch, but we also provide regulatory support because of our global footprint. As you can see on this slide, we're serving over 70 countries through 19 different sites. So while our headquarters are in Italy, outside of Venice, we also have an analytical center in Boston, various locations in the US, and across multiple continents. So diving deeper into why we're here today, we wanted to look at some of the main trends that are happening in the biologic market segment. It's one of the largest segments with expected annual compound growth greater than 7% through 2027. And because of this patient centricity, there's a lot of move from hospital care to home care, which is also driving the adoption of various drug delivery systems like auto-injectors, pen injectors, and on-body wearables. And then the formulations of these drug products are increasingly complicated due to the use of syringes or auto-injectors to deliver into the subcutaneous space. Drug manufacturers are limited to dose sizes in the 1 to 3 milliliter range. So if they want to deliver more medication, they basically need to increase the concentration of the drug, which is leading to higher viscosity formulations, which are more challenging to deliver. So 4 solutions that we'll be talking about today are what are some early stage analytical services to help facilitate the selection of the right container, especially since the prefilled syringe market has grown rapidly due to all of these previous trends. The second thing we'll be talking about what are some breakthrough solutions to mitigate specifically silicone challenges, one of the bigger challenges for biologics and the intravitreal space. Third will be the syringe design for patient-centric auto-injectors, which is how can these syringes be more compatible for auto-injectors that can improve the patient experience. And finally, the 225 syringe format and special thin wall needles to overcome the viscosity challenges that I was just mentioning with these higher concentration drug products. So diving into what early stage analytical services are. We're focused in this development space, pre-commercial generally, in the preclinical through phase 3 of various drug trials. And this could start with just having drug product stored in a vial, or you may have goals to ultimately commercialize a product that's stored in a prefilled syringe or a special drug delivery device. The goal is to be involved in each of these stages as early as possible in order to better de-risk and understand the boundaries of your product, both as a drug product and in these primary containers or drug delivery devices. Our goal is to follow your molecule's life cycle as a development and supply partner. Some ways that these support can come in the form of is head-to-head comparisons or engineering design verification. These are taking, be it our product or other products that are available on the market, and testing them with your specific drug substance or drug product to see if there's an optimal solution or a confirmation that multiple solutions are available, and you can choose which makes the most sense for you based on your own drug product or commercial supply requirements. Additionally, both US Tech, our Boston location, and EMEA Tech, our Italian location, have ISO certifications from 9001, 1345 to 17025. The types of services that we provide range from silicon coating analysis and subvisible particle analysis to more systemic, like container closure integrity methods or testing methods, not for human use fill finish capabilities, and then finally functional container and device testing. Keep in mind that the journey to commercialize a product has many stages. So from feasibility to commercialization, there's development of both the device as well as the test methods needed to verify and validate the quality of those products. An example of how we may work with you is that if you were to provide the drug substance and either other containers, or we can provide our own containers as well as the closures, we can assemble them all together through our fill finish process, subject them to any necessary conditioning, so environmental aging, such as at an elevator temperature, or mechanical stress like transportation or shipping simulation, drop tests, etc. And then finally conducting a entire battery of subvisible particle testing, container closure integrity testing, and other mechanical tests that may illustrate the compatibility of the drug product with the container. Oftentimes, drug companies are focused on the quality of their drug product, but maybe are less familiar with how the drug container or the device may degrade or change in performance over time. Our expertise helps confirm that both sides of the combination device are in a good position over the shelf life of the product. So now moving through to talk a little bit more about the silicone challenges specifically. So across these three different sectors in terms of sensitive biologics like monoclonal antibodies, deep cold storage products like the mRNA products, very similar such as the COVID vaccine, and intravitreal products, a common theme are silicone sensitivity or sensitivity to subvisible particles. So for example, in the intravitreal situation, there are just simply regulatory requirements that are more strict on the quantity of subvisible particles. The reason why these silicone particles are a concern is because they can interact with the drug product or the drug solution. These could cause protein-based drug aggregation, loss of potency or the efficacy of the drug product, or worse, in terms of harm to the patient, there could be immunogenicity or toxicity, which as safety concerns may result in loss of the efficacy of the drug product or needing to switch to a different therapy. How these issues come about is basically the interface between the drug product, any air in the container, and the silicone layer that helps lubricate the motion between the plunger and the syringe barrel. As different proteins may adhere or absorb to the inner surface, they may be displaced by the movement of the air bubble through mechanical actions such as shipping or being dropped. And then these can further aggregate as they become a surface to nucleate larger particles inside the drug product. So we'll talk about two methods to study this phenomenon. One is light obscuration, and the other one being microflow imaging. In both of these cases, we're talking about the micron up to the tens or 100 micron range, but various USB standards like 787, 7088, 789 are very focused in the 10 to 25 micron range. One of the reasons why silicone is being talked about so much in this is because of this focus on that it is the most common contributor to the subvisible particles. As you can see on this graph here, the vast majority of the particles detected are subvisible particles made of silicone oil. While there are other particles like proteins that may have aggregated or remnants of proteins or contaminants like glass flakes or rubber compounds, what the silicone oil droplet looks like is what is the most common subvisible particle. The other aspect from our testing experience is seeing how important it is for the mechanical conditioning as well as the drug formulation to be accurate when doing these studies. So for example, drop or vibration helps exacerbate the amount of particles that are formed, but are also representative of what you may expect of products that are shipped around the world or that are delivered from the manufacturing site to the customer. The other aspect is that drug buffer formulations typically use a excipient called polysorbate, which helps prevent the formation of aggregated particles, but it increases the quantity of silicone oil particles, which then has a negative effect on the overall quality of the drug product. So at this point, I'd like to introduce Enrico Barichello, who will go into some solutions to all of these challenges that we've been talking about. Hopefully, he'll be able to allow some more clarity as to the technologies that can prevent these problems.
Thank you, Alan. And now I would like to focus on the Alba platform, which is one of the solutions that helps to mitigate the silicon challenge risk. And so for this reason, Stevanato Group has recently introduced this type of platform, which consists of a broad range of syringes in different formats. And this type of technology rely on a syringe that has a specific coating, which is a cross-link through a plasma treatment. This type of plasma treatment helps to provides or to create more robust connection between the glass and the silicon layer, and this presents benefits in terms of reducing subvisible particle amounts, as well as maintaining exceptional gliding force over the shelf life of the product. This type of coating is an hydrophobic layer, which is extremely resistant also to challenging conditions. And we will see also later some of the tests that is being conducted to demonstrate its resistance to aggressive conditions. In this slide, indeed, we would like to present some of the study around the subvisible particles test through light obscuration method, as well as microflow imaging, up to 6 months stability. The syringes were filled with a placebo solution containing also polysorbate in 0.03%, and this somehow even stressed more the container in terms of silicon particle release, as well as the container were also stressed through a mechanical agitation before testing. If we see the light obscuration test at the storage condition of 40 Celsius degrees and 70% raw humidity, which is mimicking also an aging conditions, which in a real shelf life can be of many months, like 15 to 18 months, we see that the Alba syringe presents an amount of subvisible particles in absolute value, which is significantly reduced compared to the conventional silicone oil syringe. So as you can see, the Alba syringe present values that are below the USP 789, which is the reference for the ophthalmic application, while for the conventional syringe, the amount is somehow above 3,000 particles per syringe, per ml. This is valid also for the time 1, so after 1 month and after 3 months. The syringe has also been tested through microflow imaging to understand if the nature of the silicon particles or the particles released were from silicon or from other contaminants. And also at 25 Celsius degrees at 70% humidity, as well as minus 40 Celsius degrees in the deep freezing, we see that the amount of subvisible particles present in the Alba category is extremely reduced, almost near to zero level compared to the conventional silicone oil syringe. So say that we have seen that the syringe can withstand different type of conditions, even the worst one, and still presenting a low amount of subvisible particles. Moving now on the gliding force test results and understanding also the stability of the layer, we have here reported some of the outcomes regarding the extrusion forces. So we compare the extrusion forces of the Alba syringe versus a conventional silicone oil one at the condition of 40 Celsius degrees and 75% raw humidity. In this case, Alba present a consistency in terms of values over time, with also small deviation, versus the conventional silicone oil, which increase the values and the variability over time, meaning that the siliconization of the silicon layer has somehow been affected by the placebo. Still, in this case, we used a syringe filled with placebo, containing also polysorbate 80. Indeed, on the right side of this slide, you can see some pictures that present the stability of the layer. This through direct microscopy evaluation, where we studied the morphology of the coating. From T0 and T6, you can see that the picture of the alba coating remains the same. So the morphotypical morphology is kept through these months of storage, while for the standard silicone oil layer, first in T0 there is the presence of the silicone that will cover the inner surface of the syringe, and after 6 months, there becomes visible some rows, some lines in the background, which refers to the glass that become visible since the silicone has been detached by the placebo. So a reason why we're seeing an increase in force over time was also due to the fact that the silicone has been somehow modified or impacted by the interaction with the solution. In summary, we suggest the ALBA platform for different biologics segments. We see that there is a strong benefits in the biotech space, thanks to the fact that the layer stability helps to have consistency in terms of functional delivery of the drug throughout all the shelf life of the product. But also, it presents, in terms of benefits, an ultra-low release of subvisible particles, which prevent to have interaction between the drug and the container. So this helps to minimize protein aggregations or reduce the efficacy and the safety of the product. For the mRNA space, we have seen that the syringe can also withstand deep cold conditions. So also the layer withstands this type of application. And we have seen that also from subvisible particles, which can be also a point of risk with the interaction with the lipid nanoparticles, ALBA presents an inferior amount of residuals compared to a standard silicone oil syringe. Lastly but not least, ALBA is widely used in the ophthalmic space, specifically in the intravitreal, where there is the need of following specific guidelines such as the USP 789, due to the fact that it answers and responds well in terms of ultra-low subvisible particles. Going now to speak about the syringe design for patient-centric auto-injectors, I would like to present the key consideration to make a syringe properly fit a device from the beginning. When we deal with the auto-injector, there is not one syringe that fits all, but there are some considerations that help to prevent any risk of incompatibility later on. First of all, we have seen that having a enhanced flange shape with improved tolerances helps to fit the geometry of the carrier of the auto-injector. We have seen also the cosmetic quality of the product and also the dimensional accuracy of the syringe can also have been a factor of making a successful integration. Specific tubes sourcing with improved tolerances is also another key point, as well as having a broad range of selection in terms of needle shield. Lastly but not least, it's really important to make sure that the syringe resists well to mechanical strength and to mechanical stress provoked by the auto-injector. So, keeping in consideration all these points, we'll make sure that your product can be successfully launched and timely launched into the market without major risk. Going to the deep of the syringe design, we would like to highlight here the main aspects in order to make sure that the syringe is compatible with the device in the early phase, specifically related to the dimensions and tolerances. Here we report the two main aspects. The L1, which is the distance between the needle tip and the upper part of the flange, and the L2, which is the needle tip distance towards the shoulder, which is the part in contact with the carrier of the auto-injector. This specific length and measurement can change according to the device, which has been selected by the final customer. What Stevanato Group can provide is a specific 100% control in line according to the customization, according to the final auto-injector. So this measurement can be modified according to the final use. Secondly, but not less important, is to make sure that the syringe provides consistency in terms of injection forces and injection time. So a first step to mitigate this type of issue is to well design a syringe, starting from the well distribution of the silicone coverage inside the inner surface of the barrel. This has been done during the development and during the validation verification of design, while also bring to the commercialization through the manufacturing controls that are present 100% in our lines. In doing so, the camera can control where there are droplets or accumulation of silicone and automatically reject the syringes that are not in line with the defect evaluation list. This helps, as you can see on the right side of the graph, to respect injection time and to not have changes in terms of, injection time over the shelf life of the product. Another key aspect is the mechanical strength of the syringe. Indeed, when there is the activation of the device, of the auto-injector, there is a stress generated to the different parts of the syringe, especially on the flange side, on the shoulder side, and on the barrel side. So the three key aspects or point of attention for the syringe supplier has been evaluated. And here we report a comparison between a test on this specific side of the syringes, comparing the standard syringe, conventional syringe, versus an improved syringe for auto-injector. Thanks to the controls that has been put in place in our manufacturing strategy, as well as reduced no glass to glass and no glass to metal contact, and reduced generation of flows, the outcomes is that we were able to double almost the resistance of these syringes. So thanks to the development conducted so far, this was one of the achievements that offer and let us to offer to our customer a key solution when we speak about auto-injector. Another key aspect to be considered when designing a syringe is the type of viscosity of the product. And we see that there is a new generation of biologics that present a high viscosity. So we would like to mention in this chapter which are the strategies to make sure there is one syringe compatible with high viscous products. First of all, there are no one only syringe strategy, but there are many strategies that can be followed. And what we have evaluated is to be focused on specific parameters of the hydrodynamic force, which is the needle length and the needle radius. So among, let's say, the possibility to increase the diameter of the needle from also external perspective, as well as increase the solution volume to have a bigger formats, or as well choosing different delivery solution, we will focus, we want to move on a strategy towards increasing the needle internal diameter, as well as shortening the needle length. Indeed, these two points of improvements was mainly to reduce the syringe needle from 12 mm, which is the standard for subcutaneous injection, to 8 mm, as well as increase the internal diameter while keeping the same external diameter of standard thin wall needle, as per ISO 9626. And in this way, we were able to introduce a new type of configuration, which is called 8 mm special thin wall. This needle is apply to Nexa and Alba syringe platform. And indeed, it has been designed for specific high-viscous biologics drugs. Looking to the benefits of choosing a shorter 8 mm needle, here we represent one of the analytical model of extrusion force for a given extrusion time. In this slide, you can appreciate the difference of choosing an 8 mm while considering a 12.7 mm, which is the standard. And you can appreciate that the difference increases linearly at the increase of the viscosity. Moving now to the injection time, so from injection force to injection time, we have also evaluated what does it mean choosing a specialty in wall needle, so an optimized inner diameter needle versus a thin wall from a delivery perspective. This has been conducted through a test that considered also an auto-injector. We considered 2.25 ml as a representative of format for high viscous, and large biologic products. And we have studied the behavior of the syringe in a different viscosity. So in this slide, you can appreciate that the fact of using a special thin wall needle can accommodate higher viscosity compared to a standard thin wall needle, and also having the possibility to stay within the 15 seconds at 50 centipoise, which is the historical limit, the historical threshold given by the industry. The special thin wall needle is offered in a different configuration, not only in the 27, but also in the 25 and also in the 29 gauge, and is fully validated with Nexa and the Alba quality. And with this, I will hand over to Alan to present which are the potential strategies that customers can consider and how we can help on this to understand the predictable model of a specific injection time of a specific syringe.
Thanks very much, Enrico. You go on to the next slide, please. So in order to answer this question about how can we make a detailed or product-specific model, we looked at things like the needle, the drug viscosity, the auto-injector spring, and how can we optimize this for the patient experience. Generally, by doing a series of plunger syringe testing, basically glided force tests, we're able to characterize the friction force between the plunger of choice and the barrel of choice, and then basically understanding typical fluid dynamics of what the diameter reduction is coming out of the needle gauge chosen, we're able to make a predictive model as to what the injection times are going to be based on these inputs. So again, that's what are the aspects of the needle, the drug properties such as its viscosity, and what are some spring parameters that the auto-injector manufacturer can provide. Next slide, please. Here you see a summary of how we compared two different springs and two different solutions. And we're able to see that the overall difference between the experimental and the simulation results in general about as good as basically negligibly different, all the way to, let's say, less than 5% difference between the two. But you can see that with the 5 centipoise solution, you can see the delivery time is on the order of 15 to 16 seconds, whereas with the 13 centipoise solution is on the order of 40 seconds, which is much too large compared to the 15-second threshold that Enrico had talked about. So using this model, we could have predicted that you would need either a lower viscosity, like the 5 centipoise solution, or possibly even different spring mechanisms that were tested here, because excuse me, the 13 centipoise solution simply would not be compatible to deliver a 15-second injection with spring 1 or spring 2. So now I'd like to turn the slides over to conclusions. So through this webinar, some things that we've talked about are the trends of how important biologics are. A lot of the problems that we've been discussing are focused on sensitivities to biologics like mAbs, mRNA, or intravitreal drugs. And then because they're often being used in auto-injectors, what are some of the functional and dimensional requirements or compatibility testing to make sure that they will work well with these types of drug delivery systems? And then finally, what are some solutions to address the increasing concentration of drug formulations, namely due to keeping drug delivery volumes low in the 1 to 3 milliliter space? So the testing that we've discussed, or the analytical services that you could evaluate, are these things like the subvisible particle count, the morphology to identify if it's silicone oil or if it's something else. There are things like the injection force and how consistent it is, the glass robustness and its consistency, and then the overall injection time or maximum injection force. There's many different products on the market, from conventional to these name brands Nexa and Alba, and ultimately it's a choice of the pharmaceutical company which is the most appropriate for their drug product needs and what are their overall product requirements. It's through these tests that you can make an informed, data-driven decision. And we hope to be partners with you in order to help you find what's the right product through the variety of options that are on the market. So at this point, I'd like to turn it back over to Scott. Thanks very much.
Thank you so much, Alan and Enrico, for some excellent content. So we have received numerous questions from the audience, which we might not have time to address them all, but let's start with the first one. So how does this help the drug product? So if Enrico, I'll hand that to you.
Yeah, sure. So thanks, Scott, for the question. The I would say, outcome that I would like to pass through this presentation is the fact that Alba can be really a solution that helps pharmaceutical companies in having, let's say, facilitated the introduction of new molecules, new products into the market, thanks to the fact that we see from a market trend that there is an increasing growth of high concentrated products. These products are very challenging in terms of making them stable in a syringe specifically. So there are a lot of content like chemical substances like surfactant and excipients that are put inside, and this type of elements can somehow interact with the container, with the syringe. So we have seen that we have tested the Alba in a different condition, even in a worst case, to really understand what happened if there is this challenges condition also. It really happens with the final product, and what happens then to the final, let's say, developers. And we have seen that the benefits that brings in terms of reduction of subvisible particles, but also in terms of keeping the injection time very consistent in the overall, let's say, shelf life. This we believe that this is the new paradigm that can really help to have more concentrated drugs, more stable, and without the risk of having less functionality risk, especially in the delivery of the solution integrated without injector. So we really believe that this helps drug formulators to have a continuous solution that make their life easier.
Excellent. Thank you, Enrico. So moving on to question 2. So how can we get this to market easier?
We can, of course, this type of product is as of today validated, so it's already present in the market as an off-the-shelf solution. And so the main consideration that has been touched by Alan during this webinar, so making sure there is the selection of the container, but also a sort of risk assessment in the compatibility between the drug and the container in the early phase of the development. This really helps to anticipate possible risk that might, let's say, arise later on. But especially, I was also consider that with the specific Alba solution as well as Nexa, really are providing the best-in-class technology. So we are already in advanced, let's say, in the field of the advanced solutions that already have mitigation strategy, especially regarding silicone oil, residuals of tungstens, glue, leachables, as well as also for the all the parts of mechanical resistance and dimensional perspective for the integration with devices. So these are the work of Stevanato Group that did in the several years, in the last 10 years, that are concentrated of technology that is already proven and delivered to the market as a key solution for biologics.
Excellent. Okay, so moving on to question number 3. So I already have a commercialized product. So how can I leverage these products or technologies?
Sure. So I think part of this is going to be driven by the comparability or compatibility testing. So if you already have a product that's on the market, it's these types of tests that can help justify to regulatory authorities that the product is either comparable or better to what is your existing commercial product. Doing accelerated stability studies in addition to real-time stability studies are going to be part of the method to address those concerns. Enrico, is there other comments you'd like to add about the Apple platform?
No, I think you well covered all the topics, yeah.
Okay, so let's move on to question number 4. So I'm having issues with visible particles. Are these also related to silicone, or are they something else entirely?
Sure. So with visible particles, this could be a different beast. And part of this could be, on extreme scenarios, it could be glass delamination, or it could just be some other kind of foreign contaminant that's accumulated into the drug substance. Some of the approaches that we do are FTIR or SEM-EDX based, which can help identify what the compound or elemental structure are for these particles. So we could filter them out and try to analyze, does it match with cotton, natural fibers? Is it some kind of metal from handling equipment? Or is it some kind of organic compound that's formed from the drug substance itself? Ultimately, doing these investigations to identify what it is, this chemical analyses are certainly within our wheelhouse. And one of your first steps to try to identify what it is, and then going through a root cause investigation to figure out where throughout the process it may have come from, be it the manufacturing process, some other kind of container interaction. As I mentioned, delamination is actually becoming increasingly relevant for customers that have high pH or phosphate buffered solutions. Having a detailed root cause investigation, something that we can be a part of, would be one of the first steps to trying to track down that root cause.
Okay, great. So moving on to 5. So I don't see subvisible particles yet, but what are the other ways that I can check the compatibility earlier on?
Sure. So accelerated aging as well as mechanical, basically shaking or dropping, or end over end forms of agitation can increase the number of subvisible particles or accelerate their formation. These are methods that we've incorporated when doing our testing to make sure that we get either a worst case or an accelerated simulation of the shelf life of the product. So even if you don't see it today, after you go through these types of conditioning or accelerated aging, that may illuminate more of what the count or types of subvisible particles you might encounter at the end of shelf life or beyond the end of shelf life, to just make sure that your product stays integral as well as is not interacting with its container over this, could be multi-year process.
Excellent stuff. Thank you, Alan. And this will be the final question. So number 6. So what does a typical TEC engagement look like?
Sure. So I think this simply starts with a conversation, and part of this is understanding what your goals are or what your problems you're facing right now. This has ranged from maybe a design verification test is not going according to plan, you're getting some failures, or it could be you're very proactive, and this is the best case, which is you are in the process of finalizing your formulation, and you're trying to figure out what are the containers that it should be stored in. Doing these early surveys, reaching out to different product suppliers can help leverage their experience as well as to testing houses such as ours to understand what are some pitfalls, what are some problems that other customers may have faced, and how you can learn from those experiences. So those types of discussions are what they would start, and then they would move into an actual formal proposal, a test plan of how we would approach your specific testing challenge, and then we'd go from there.
Excellent. Well, thank you, Alan and Enrico, for such insightful content. I think we've all really enjoyed it. We have reached the very end of the webinar now, so we appreciate all of the participants' engagement as well. And apologies to those whose questions couldn't be addressed, but the Stevanato Group will be reaching out to you as soon as possible with answers to those. So Alan, Enrico, any final words for the audience today?
Sure, I'd just say don't be a stranger. If you have any questions about the products or test methods that we talked about here today, feel free to reach out through our website, and we'd be happy to have a deeper conversation with you. Enrico?
Thank you all for attending this webinar, and we look forward also to receiving your further question, if any.
Thank you so much. Okay, we'll end now, but again, thank you to the audience, and we shall see you soon.